everybody. Today I am going to talk about the cloning vector PBR322. Okay. So the topic here is biotechnology principles and processes. In biotechnology, we are using so many tools. The first and foremost tool is the restriction enzymes. The second very important tool is the plasmid, which is acting as a vector. The vector what we are going to use here is the PBR322. So today I am going to talk about this PBR322. So if you take biotechnology, we are having so many vectors, especially for NEET preparation. As per the CBSE, they have kept PBR322. Before entering into this PBR322, you should know what is meant by vector, why we are using the vector is the plasmid here. Okay. In general, vector means what? Carrier molecule. Carrier molecule means what? From one place to another place. The transfer of the molecule is called as carrier molecule. For example, if you take the female Anopheles mosquito, which is the carrier, which is the vector for malaria, which is going to carry sporozoites, the infective form of the pathogen. Same like that, in order to carry our gene of interest, we are using the plasmid as the vector. So what is the first and foremost property of the plasmid? If you take bacteria, all bacteria do not contain plasmid, only very few bacteria contains plasmid. So what is the specialty about this plasmid? For example, this is the bacteria. This bacteria will be having the chromosome. Okay, in this bacteria, the double-stranded DNA. So, the double-stranded DNA which replicates autonomously, extrachromosomal double-stranded DNA which replicates autonomously, that is called as plasmid. Okay, so because of this property, we are going to use the plasmid and then we are going to construct as per our requirement. Okay, so that is called as vector here. To carry out the gene of interest, we are using the plasmid. What is meant by plasmid? It is an extra chromosomal double-stranded DNA which replicates autonomously. This is the reason we are using plasmid as the vector in biotechnology. Now we will say, what is this PBR322? What is the salient features of this PBR322? So in order to understand the salient features of the vector, salient features of vector. Salient features means what? See, our gene is like a treasure. It's like a more than a gold. To carry this gene of interest, the vector should have some special characteristic features. So what are the special characteristic features the vector should fold? So the first and foremost thing is oricide. The vector should contain the oricide. And the next one is it should contain multiple cloning sites. Multiple cloning sites. In short, it is called as MCS. Okay, I will explain what is that. And the third one is it should contain the selectable marker. Selectable marker. The selectable marker what we are using here is the antibiotic resistant genes. Antibiotic resistant genes. Now the fourth one is we need to consider about the size of the vector. So these are the four important points the vector should carry. So what is the first and foremost point? Oricide. So what is meant by oricide? Oricide means origin of replication. If you take any vector, it should contain one site which is called as origin of replication. For example, this is the structure of PBR322. Okay, this PBR322, as per our salient features, it contains oricide. Oricide means what? Origin of replication. So what is the purpose of this origin of replication? Whatever the gene you are inserting, okay, see our purpose is what? The vector should carry the gene of interest. Okay, for example, I am going to insert my gene of interest in this ampicillin resistant gene. So that what will happen means this gene of interest, it will go and integrate to the site of oriside, and whenever the plasmid undergoes the process of replication, the gene of interest also will get replicated. As a result, we are getting so many clones. Clones means what? Single gene in large number. So that is the purpose of oriside. So the next one is the multiple cloning sites. So multiple cloning sites means what? If you take any plasmid, any vector, it should contain so many restriction sites. Say in biotechnology, restriction site means what? Restriction